Hello and welcome to my BMI Journal Club presentation, Immunoinformatic Design of an Epitope-based Immunogen Candidate against Bacillus anthracis. This paper was published by Shimaki and Kobashi in Biomatics in Medicine Unlocked 2021, Volume 24, and my name is Carissa Perry. So why did I choose this particular paper? This paper is applicable to my main Bio 470 proposal as it's based on epitope-based methods of vaccination. It makes use of various bioinformatic databases, methods, and protocols, some of which we have looked at in this class. And it's also applicable because Bacillus anthracis can be used in biological warfare and has a 50% mortality rate, which makes this a constant threat to any population at threat for bioterrorism. What biological warfare is the use of toxins of biological origin or microorganisms as weapons of war. And what you can see here is a scanning electron micrograph of Bacillus anthracis. So presentation outline, I'll be looking at biological and medical problem, applications to bioinformatics, informatics background, goals of research paper, research methods, results, evaluation of methods, informatics biomedical assessment, some concerns, and a summary. So biological medical problem. So Bacillus anthracis is a spore-forming rod readily found in soil in mostly tropical environments, and it causes the disease anthrax. Now, anthrax can be caused by inhalation, cutaneous, or gastrointestinal exposure. The illness is caused by germination of spores in host macrophages, which release toxins known as edema factor, the protective antigen, and lethal factor. And protective antigen and lethal factor are the target of this specific paper. Anthrax has a high morbidity and mortality rate, and it can be used as a biological weapon. And there's evidence of it being used as a weapon in instances in 1979 where anthrax was released in Russia, which led to the death of 66 people. And in 2001, shortly after 9/11, the United States Postal Service also experienced anthrax attacks, which led to the death of 5 people. Vaccination against anthrax is significant for decreasing the damage that can be done in biological warfare, and is also significant in decreasing damage from spore ingestion from soil, animals, or agriculture. And veterinarians, farmers, and wool sorters are people at the highest risk for anthrax infection, as they're the most exposed to this microorganism. Anthrax also disproportionately affects developing countries in warmer tropical climates, and these countries cannot afford the same treatment methods available to the developed world. There's also the danger of exposure during vaccination development, as the scientists who are developing vaccines are being constantly exposed to a bacterium that can form spores and potentially make them sick. So methods to not have to use the microorganism itself are definitely looked at as promising for anthrax vaccination. In this paper, the researchers specifically target the lethal factor and protective antigen, and these proteins form complexes of lethal toxin and edema toxin. And you can see from this diagram that the protective antigen complexes with lethal factor produce lethal toxin, and also complexes with edema factor to produce edema toxin. And now these proteins have been successful targets of the anthrax vaccine, but biothrax effectiveness is limited, as this is a vaccine that requires multiple doses, and people who have the biothrax vaccination have to get annual booster shots. The new generation of biothrax uses CpG oligodeoxynucleotides, which boost the immune response of both natural killer cells and T cells. The challenge with anthrax vaccination is to find the best adjuvant. In this particular study, the researchers used Mycobacterium tuberculosis, the 50S subunit, as the adjuvant in the vaccine. Anthrax virulence is produced by 2 processes. One is an antiphagocytotic capsule made by PGA, and also the production of bacterial toxins, which is the target of this paper. The PXOI plasmid is responsible for production of the protective antigen and the lethal factor of anthrax. A protective antigen attaches to the cell exterior receptors. There's cleavage of an N-terminal protein, and this acts as a translocation factor for the lethal factor. Some applications to bioinformatics. The study looked at amino acid and nucleotide sequences, which are available on various databases. The study also looked at in silico methods based on epitope mapping and molecular docking, and those techniques will be explained further later on. And they found specific amino acids epitopes and did a mass comparison to T cells, B cells, and bacterial proteins. So they took the sequence of bacterial proteins and compared their sequences to T cells and B cells, and those sequence overlaps are referred to as epitopes. Sequences with the highest immunogenicity and binding to immune cells would then move forward in this process, and it goes from targeting pathogen to extracting sequences to comparing immune cell sequences, checking for immunogenicity, allergenicity, and proposing the best epitopes. The main technique that we're looking at is epitope mapping, which determines amino acid sequences common to both the pathogen and immune cells. And epitopes can be either continuous or discontinuous. It can either be a sequence that is one section on this part of the protein and then does a jump and is another section on another part of the protein, which is known as a discontinuous epitope, or it can be continuous. So it can be one solid connected section of a protein. This paper also looks at molecular docking, which simulates molecular binding, molecular specificity, ligand binding, and factors in different rotations. And the molecular docking method looked at 70,000 or more different rotations of a protein. So these are different conformational changes that can occur in protein binding. And this study also used molecular dynamics, which factor in structural changes between proteins, ligands, and receptors. Some of the goals of this research paper I've already touched on, but the basic idea is to use in silico methods to develop a candidate vaccine for Bacillus anthracis. And by doing this, they form a hypothetical chimeric protein as the vaccine candidate using bioinformatic databases and servers. And then this hypothetical protein is checked for antigenicity, toxicity, allergenicity, which lowers the chances of side effects when it's to move into wet lab or clinical trials. And the researchers do this by predicting the structure of the chimeric protein and the binding of the chimeric protein to anthrax receptors. And then this hypothetical protein can be synthesized and cloned for mass production in a new vaccine. So the goal is to develop a potential epitope-based chimeric protein that can then be moved into wet labs and clinical trials. So the epitopes at the lowest allergenicity are considered the best candidates. And these are predicted through informatics servers comparing sequences and structures that have adverse effects. And the ones that have the least toxicity and allergenicity then move forward to test immunogenic properties and the ability to bring out an immune response. Epitopes with the best immunogenic scores and properties then move forward to predict their secondary and tertiary structures and move forward to the molecular docking. And molecular docking factors in approximately 70,000 different protein rotations. And these processes uses principle of sequence extraction with accession numbers, sequence alignment, comparison of protein structures, binding scores, and algorithmic prediction of protein structures. So to begin the research methods, the researchers targeted protecting antigen and lethal factor. They used the 50S ribosomal subunit of Mycobacterium tuberculosis as an adjuvant, and to form a hypothetical chimeric protein, which contains the successful epitopes as deemed by immunogenicity scores. And these epitopes are joined together in the protein by GPG-PG linkers. The N and C terminus also contain EAAK linkers, which increase structural stability. And the hypothetical protein also contains 6XHI to S tags at the C terminus, which increase overall protein purity. Now, this is a flowchart of the different servers and databases that these researchers used. I will be highlighting these later on, but the basic flow through consists of target antigen, extract sequences, compared to immune cells, check for allergenicity, toxicity, check immune profile, protein solubility, predict secondary structure, predict tertiary structure, move to molecular docking, and then propose the final epitopic vaccine. So these are the different servers and databases used. UniProt is a database, and it was used for the extraction of LF and PA protein sequence, and these are the accession numbers that they used. GeneBank was used in similar methods, except the gene sequence or genetic sequence, nucleic acids were extracted of LF and PA, and these are the accession numbers. BC-Pred was used to predict T cell and B cell continuous epitope extraction. CBTOPE is used for investigation of nonlinear B cell epitopes, which is also like the discontinuous epitopes. So ones that are nonlinear could be circular or jump from one section to another section. IEDB was used for investigation of T cell epitope competency. So if a T cell epitope isn't useful or isn't competent, isn't going to work properly, then this server was used to check, is this epitope actually plausible? IFN epitope was used for interferon gamma and IL-10 epitope potentiality, which are different immune cells. VaxGen V 2.0 was used for overall prediction of antigenic properties. AllerCat Pro was used to predict allergenicity. ToxinPred was used for predicting toxicity of epitopes. CIMU-IMSIM was used to predict immune response profile, so how well these epitopes actually cause an immune response. ProParam was used to calculate amino acid composition and properties. PepCalc was used to validate solubility of the protein. GOR4 was used for analysis of the secondary structure as well as Raptor X. iTaster was used for tertiary structure prediction. 3D Refine was used to refine the protein's tertiary structure. ProSA was used for further refinement and compared with pre-structured native proteins. SAVES V6.0 is used for analysis of protein structure quality. And HADOC is the molecular docking software, which simulates receptor ligand binding. So the results. A total of 32 epitopes were initially selected. 13 were linear and 19 were nonlinear B cell epitopes. Out of those, 16 MER, which means 16 amino acid epitopes, were selected after cross-matching. And these 11 epitopes were considered to have the best immunogenic properties. Antigenic properties were predicted by VaxGen 2.0. The allergenicity measured 0.852, which is a good score for allergenicity. Anything under 0.45 is not good. So 0.852 is a very good score for this. And the conformational epitopes consist close to 90% of B cell epitopes. So close to 95% are conformational epitopes. And these are the individual sequences that were extracted by comparing lethal factor and protecting antigen proteins to B cell proteins. And all these letters correspond to the different amino acids, which we've learned about in the class. The results of testing to B and T cells. T cell epitopes were predicted by MHC1 and MHC2 prediction tools. And what those are is a different immunological cells. And the IDEB server was used to check those, which simultaneously checked B cell epitopes as well. So this epitope sequence was realized to have the best interactions with both types of immune cells, which means that would be the most promising epitope to use specifically for T cell epitopes. And more than half of these epitopes also had positive induction scores for the other types of immune cells, including IFN gamma, IL-4, and IL-10. And what this figure is the hypothetical peptide. So you can see there are GPG, PG linkers in between each individual epitope, and then the EAAK linker seen at both the N and C terminus. And the adjuvant is seen at the N terminus, which is the 50S ribosomal subunit from Mycobacterium tuberculosis. So further results, so the protein itself, which you just saw in the figure before, it would consist of 382 amino acids that had a molecular weight of 39.44 kilodaltons, isoelectric point of 5.12, which means it's slightly acidic. The protein is stable, hydrophilic as indicated by gravy. The protein is also thermostable with an aliphatic index of 67.4, and it's soluble. It had a server index higher than 0.45, which means it's a soluble protein. The final protein secondary structure and tertiary structure can be seen in this diagram here, and the final protein was estimated to comprise 38.5% of alpha helix, 12.8% extended strand, and 43.2% random coils by the GOR4 server. Molecular docking showed crystal structures with the lowest resolution of proteins, or molecular docking. So results for molecular docking. Molecular docking uses crystal structures with the lowest resolution, and they're prepared for docking by removing water, unrelated residues, and heteroatoms. Molecular docking generated 30 rigid molecules for protecting antigen and the anthrax receptor, ranked by cluster size. It also generated 80 flexible models for the protective antigen and anthrax receptor, also ranked by cluster size. The best epitope for the protective antigen had a delta G of negative 10.8 kilocalories per mole and a KD of 1.2 times e to the negative 8. For the lethal factor, it generated 334 structures in 39 different clusters. And the best epitope had a delta G of negative 1.18.4 kilocalories per mole and a KD of 3.2 times e to the negative 14. And this is a graphical representative of the molecular docking. So you can see the relative molecular docking between the vaccine itself and the anthrax receptor. Now for my evaluation of these methods, I believe that these researchers were successful as they proposed a epitopic vaccine that had low toxicity and allergenicity, and molecular docking found the best orientation and positioning of the proteins to the receptor. It was also successful as they used multiple servers that acted as checkpoints. They served the same purpose, but it acts as checkpoint because when they use GOR4 and Raptor X, they could check, okay, does the sequence that I found or the structure that I found with GOR4 line up with the sequence that I found with Raptor X? And their results do not rely on one specific server or database. They made sure to use a broad range of different servers. This article does not explain, in my opinion, does not explain the significance of scores, especially in the molecular docking section. But it is promising because it's effective at showing protein receptor interactions. It's not enough to just propose a specific protein. You have to know how they interact with antigen receptors in order to predict how a protein is going to act in wet lab or in clinical settings. And the readers also left wondering what the different scores corresponded to which epitope, as in they just listed, oh, the best epitope had this score. They didn't actually list, oh, this epitope with this sequence had this score. So biomedicine assessment, it's applicable to the areas of vaccine development, protein receptor interactions, protein conformation, immunology. It demonstrates that protein vaccination is a plausible technique for anthrax infection. It also highlights the importance of having effective linkers and adjuvants. This study could benefit further from explaining why they specifically used the 50S ribosomal subunit and why they used Mycobacterium tuberculosis as a specific source. And it also brings promise to future vaccine development and promise to move into wet lab experiments that if this hypothetical protein gives us these results, then it's likely in a wet lab, it's also going to give us promising results. And bioinformatics wise, it uses a wide range of databases and it has lots of checks and balances. It also uses information in both protein and nucleotide settings and uses a wide range of servers, including allergen and toxicity servers. These servers are readily available and they can contribute to determining structure of other unrelated proteins such as enzymes, substrate interactions as seen in biochemical reactions. And the Haddock server especially is promising as it uses molecular docking to determine how proteins interact with their different receptors and can also be used for enzyme and substrate interactions. One downside is that the scoring system can get confusing and the paper would benefit from further explanation of this scoring system. So some of my concerns, the paper had a good layout. Significance of scores could also be explained further. And I was wondering, will the proposed vaccine actually follow through on predicted allergenicity or toxicity, or depending on random events, could it potentially cause problems for some individuals? This protein is also theoretical, and it may be difficult to achieve in wet lab experiments and wet lab settings. How does it factor in unpredictable events? Are those factors into these algorithms that the researchers used? And I know I said it was good that they used a multitude of servers, but it could also get confusing. And what I was wondering, is there one program or one server that factors in all the benefits of using these multiple servers, but yet is like a one-stop shop for this type of project? So in summary, it's important to combat anthrax, which is a spore-forming bacillus, as it disproportionately affects developing countries and poses a threat as biological warfare and potentially found in soil and agriculture. This paper proposed an epitope-based immunogen candidate. It tested two immunogenic cells and used bioinformatics servers to get sequence data and determine potential epitopes with the most sequence overlap. The paper determined 11 possible epitopes, all joined to the same chimeric protein using GPGPG and EAAK linkers, which showed minimal toxicity and allergenicity. Similar methods can also be used for vaccinating against other antigens such as HPV, which causes cervical cancer. And other conclusions could find different possible epitopes to use in this vaccine, or if they change the protein target from LF and PA to different proteins, it could be promising in other areas of anthrax infection, not specifically just with those proteins. They can propose epitope-based vaccines for other proteins of anthrax that may be more effective than using LF or PA. And these are my references and further reading. Thank you.